The 2013 Albert Lasker Basic Medical Research Award for discoveries concerning the molecular machinery and regulatory mechanism that underlie the rapid release of neurotransmitters. Honors Richard Scheller and Thomas Südhof.
The brain is responsible for our abilities to think, to feel. The brain's responsible for our ability to learn and remember. It's responsible for everything we've done from the moment we're born to the day we die.
The independent discoveries of Richard Scheller and Thomas Südhof have provided an understanding of the system by which nerve cells release the chemicals they use to communicate with each other.
And it's this massive communication between one nerve cell or thousands of nerve cells communicating with thousands of other nerve cells that all of us who are scientists who study the brain believe is the fundamental process that allows our brains and the nervous system in general to mediate all these amazing functions.
The system involves balloon-like containers called synaptic vesicles, which hold chemicals called neurotransmitters that carry messages from one nerve cell to the next. When triggered by an appropriate signal, these vesicles fuse with the nerve cell membrane to liberate their contents and thus pass along their message.
Their goal was to solve that molecular machine. Find the proteins that comprise that molecular machine, figure out how these proteins interact with each other, and then figure out how they actually work.
In the 1980s, Richard Scheller, trained in the rapidly evolving field of molecular neurobiology, initiated his quest. Now, over 20 years later, he recalls his fascination with the brain and how it works.
This human brain is comprised of neurons that make approximately 10 to the 15th connections with each other at sites called synapses. So we have 10 to the 15th of these little complicated machines all firing and working in our brain in ways that are allowing me to have this conversation and to remember what happened when we made these discoveries 20, 30 years ago.
Initially, Scheller focused on the synaptic vesicle proteins. The first one he identified, he called vesicle-associated membrane protein 1.
VAMP1, which turned out later to be the first vesicle snare protein on the vesicle that is directly involved in the membrane fusion. So this is the first molecular link.
After isolating VAMP1, Scheller went on to discover a brain protein he named syntaxin.
Syntaxin was on the plasma membrane. So now we had a link, a physical link between the vesicle and the plasma membrane. And VAMP and syntaxin turned out to be the first V snare and the first T snare, two of the proteins that are directly involved in the membrane fusion process. Each of the red dots is a synapse.
As more information was gathered, the interactions among these proteins and the way that those associations might instigate fusion became clear.
As it zippered.
Subsequent investigations supported a scenario in which the proteins zipper together, forcing fusion.
I'm in a little bit of awe that, given where we started over 30 years ago, that we figured this out. But that's the fun part. That's the great part of all this, that we did figure it out. I am in awe of the beauty of how it works.
We are basically trying to complete what we started.
How neurons communicate had been an interest of Thomas Südhof since his days as a medical school student.
Here are the neurons, you can see them here. And I decided to do science, and I decided to do what interested me most, which was to try to understand how neurons communicate.
For his research purposes, Südhof defined the problem and what questions he felt must be answered in order to understand the molecular underpinnings of neurotransmission.
The problem that I was interested in is how are synaptic neurotransmitters released at a synapse. that was what I was interested in. And that problem really is composed of three major questions. One is how synaptic vesicles fuse, because they have to fuse in order to release. The other one is, how does calcium actually trigger that fusion, which is amazing as a process because it is so fast and exhibits a high degree of plasticity. And the third question is, how does a synapse manage to localize calcium influx right to where the vesicles fuse? So these are the three questions.
The first protein Südhof isolated he called synaptobrevin, which was the same protein as VAMP1. This was followed by isolating another vesicle protein he called synaptotagmin. He later showed that it binds calcium.
It senses the calcium and translates the action potential signal that allows calcium to flow into the nerve terminal into an exocytosis signal by binding the calcium and triggering the fusion, and thereby triggering exocytosis.
Südhof then further elucidated the mechanism of regulation with the discovery of the protein COMPLEXIN and demonstrated its essential role in calcium-regulated neurotransmitter release.
And these two proteins are synaptotagmin and an adapter protein called COMPLEXIN. So these proteins, and what happens is that synaptotagmin has domains that bind calcium, and when they bind calcium, they change the way how they interact with lipids and with snares. And this COMPLEXIN molecule helps with that. The nerve terminal is full of synaptic vesicles. There's hundreds of them.
For over 2 decades, Südhof's ongoing research has addressed the molecular mechanism of neurotransmitter release, unraveling many of the mysteries of one of the brain's fundamental mechanisms.
Understanding how a synapse changes, how a synapse computes a presynaptic signal into a postsynaptic response will become much more of interest in future and will become much more in the focus of research in future.
By systematically exposing and analyzing the proteins involved in neurotransmitter release, Südhof and Scheller have laid the foundation for future research, which will one day help in understanding the defects contributing to many diseases of the brain.
As scientists, we all believe that if we can understand the pathophysiology, the causes of these diseases, that's the best path forward to finding much better, more efficacious treatments.